What is dark reaction? Reaction which require darkness? Sorry, that is not the correct answer. Dark reaction is those reaction in which actually light is not required. So it can occur in both day as well as the night. This is Dr. Pankaj Kumar, your mentor, and in today's sessions we will be talking about the dark reactions or the Calvin cycle, right? So you see, the dark reaction occurs in stroma of the chloroplast. It was worked out by Calvin because actually he was working on an alga chlorella, and what he did, he has used the technique what we call autoradiography. So in case of autoradiography, what happens that radioactive material is used, and after a certain period of time the product is taken out and it is analyzed. So on the basis of his autoradiography technique, he worked out the entire cycle, what we call it is the Calvin cycle. Okay. So in case of Calvin cycle, what we see that the assimilatory powers that is formed in light reaction in the form of ATP and NADPH2, these are going to be utilized, and after this the CO2 or the atmospheric CO2 is going to be fixed in the form of carbohydrate. So it occurs in certain steps. We will talk about those steps. So for dark reaction, it can be divided into 4 steps. What are they? They are first the carboxylation, then we have a phosphorylation, then we have a reduction, and followed by the regeneration of the CO2 acceptor in the form of ribulose biphosphate, right? Let's talk about the first thing, and the first thing is the carboxylation. Now in case of carboxylation, what happens that 5 carbon compound ribulose biphosphate act as a CO2 acceptor. Okay, so they bind with the CO2 in the presence of enzyme what we call Rubisco. Now Rubisco is the most important enzyme. This is the most abundant protein on this earth, and as per one estimate, 16% of the total protein constitute the Rubisco, right? So what happens that RUBP binds with the CO2. As a result, they will form a 6 carbon unstable compound, and they soon split into 2 molecules of 3 carbon compound what we call 3-PGA. Okay, since the first stable product is the 3 carbon compound, so this is a stable product. Okay, so since the first stable product is a 3 carbon compound, we call Calvin cycle as a C3 cycle as well, right? Now let's see, once the 3-PGA is formed, our next step is the phosphorylation. In case of phosphorylation, the assimilatory power ATP that was formed in light reaction, that is actually being utilized over here. So what happens? In the presence of enzyme kinase, the 3-PGA is actually getting converted into 1,3-di-PGA, right? So that is the product formed after phosphorylation. Okay. Now you see, this is an acid. Our next reaction is a reduction. So if acid getting converted into aldehyde, it is reduction, right? So what happens here? That 1,3-di-PGA is reduced to PGL, and here the second assimilatory power in the form of NADPH2 is actually being utilized, right? And in the presence of enzyme dehydrogenase, NADPH2 is actually being formed. Okay, so you can see there 1,3-di-PGA plus NADPH2. In the presence of dehydrogenase, the phosphoglyceraldehyde is formed. Now this phosphoglyceraldehyde is going to act as the material which will regenerate number of molecules, right, intermediate molecules of the Calvin cycle, and eventually the glucose is fixed and the CO2 acceptor in the form of ribulose biphosphate is formed. Okay, but before that, one important component is formed what we call RUMP, and ribulose monophosphate is again going to utilize 1 molecule of ATP, and thereby the RUBP is going to be formed, right? Let's see. So we can show there as like that PGL after a series of reaction they will form a fructose, and fructose during night actually gets converted into starch. That is the usual conversion. Okay, so we know very well that during day hours it is converted into fructose, and during night hours actually it's converted into starch. Now this is in the process they forms another component what we call RUMP, that is ribulose monophosphate. Okay, so this ribulose monophosphate, again with the help of enzyme kinase, right, they form ribulose biphosphate. Okay, so here again kinase enzyme is there, right? So ribulose biphosphate, and this is the again CO2 acceptor. So this CO2 acceptor is generated, and the cycle thereby keep on going on. So this is all as far as what we call the Calvin cycle. Okay. So before we conclude, let's talk about the energetics, that what are the energetics that is involved over there. Okay. So here what happens for the fixation of 1 molecule of CO2, right? What we need 3 ATP and 2 NADPH2. So just recall what are the places where 3 ATP were used. You see, the first place was the very conversion of 3 PGA into 1,3 di-PGA, right? It was a 2 molecule, okay? So thereby here utilization was a 2 ATP, right? And the second was that the RUMP is actually getting converted into RUBP, okay? And here 1 ATP is being utilized. So thereby altogether 3 ATP is required for the fixation of CO2, right? And what is the site of NADPH2? You see, 1,3-di-PGA getting converted into PGL. Again, it is a 2 molecules, so 2 molecules of PGL form. So here also what we see that NADPH2 is actually being utilized. Okay, right? So since for 1 carbon or 1 molecule of CO2, 3 is required. So naturally for 1 glucose, we know very well in glucose there are 6 carbon. So that's why the requirement is 18 ATP and 12 NADPH2. For more important thing that you should keep in your mind, that for the fixation of 1 carbon, Calvin cycle has to run 1 time. For the fixation of glucose, how many times the Calvin cycle has to run? Answer is 6. So many a time this question is asked, one particular organic molecule will be given over there, and the question is asked how many times the Calvin cycle has to run. For instance, suppose if the question is asked that for forming a molecule of 3 PGA, how many times the Calvin cycle has to run? Please remember, it is talking about fresh 3 PGA, not the internal PGA that is already present in a plant. So for fresh 3 PGA, 3 carbon is required. And for each carbon, the Calvin cycle has to run 1 time, isn't it? So that's why the answer to this particular question is 3. The Calvin cycle has to run 3 times. Okay. So this is all as far as entire Calvin cycle is concerned, right? Thank you.